welcome to the next knowledge clip of thermodynamics. In this knowledge clip, we are going to talk about redox couples in electrochemical cells. Chapter 6 deals with the thermodynamics of redox reactions. Last knowledge clip, we already got to know an important redox reaction, the reduction of NAD+ to NADH. This happens in the citric acid cycle, which generates multiple NADH molecules that are then used as electron energy carriers so that they can be oxidized again in the next step, for instance, to generate a concentration gradient of protons across the inner membrane of a mitochondria. So this is an example of an electron transfer reaction, but it's quite complicated, as you see here. And the most simple way to actually illustrate such electron transfer reactions is with metals and their corresponding salts. So here a metal plate, zinc, out of zinc, is submerged in a solution of copper sulfate in water. So if you put copper sulfate in water, it forms two ions. Because it's a salt, it forms copper 2+ ions and sulfate 2- ions, which now will be inside of this solution. At the electrode, the zinc metal will actually begin to dissolve into the copper sulfate solution by forming zinc 2+ cations. 2 electrons are formed during this oxidation that are now available for another step that comes next. But first, let's look at the equation that describes the oxidation of zinc, where it goes from a solid to a solution state upon releasing 2 electrons. So that's an oxidation reaction where electrons are taken from a metal, for instance, so that they are made available for a next reaction, which is a reduction. The reduction happens with the copper 2+ cations that are in this solution. They will actually pick up the 2 electrons and therefore form copper, which is a solid, and it basically deposits onto the surface of the zinc plate. So the reaction equation for this is given here. The 2 electrons from the zinc are going to the copper 2+ ions that are in solution and form solid copper. So that's called a reduction reaction, where electrons are added to a molecule or to a salt cation here. So both of these reactions together are called 2 half reactions, right? Because only together they form a redox couple. So the electrons, they go from the zinc in solid to the copper 2+. And the solid zinc is oxidized while the copper 2+ is reduced. So now let's do the opposite experiment. Let's take a copper plate and submerge it into a solution of zinc sulfate this time. So the opposite than what we did here. If you look at the copper plate, you would think that now the copper can as well go into solution, forming 2 electrons in the copper 2+, and those 2 electrons would then be made available via this reaction equation to the zinc 2+ that's in the solution, so that solid zinc is deposited via this reaction. This is not going to happen. Zinc is not going to be reduced, and copper in this example is not going to be oxidized. Why is that? It's because the oxidized form of zinc 2+ has a lower free energy than the oxidized form of copper 2+. Therefore, it's energetically more favorable for the zinc 2+ to stay in solution and for the copper to stay as a solid. That's why this is not going to happen, but that is going to happen, because forming zinc cations produces a lower free energy state than the copper 2+ cations. They instead become a solid by becoming reduced. So copper attracts electrons more strongly than zinc. So how can we measure this Gibbs free energy difference between the copper 2 plus and the zinc 2 plus cations. It turns out that that free energy difference can be related to a voltage in a so-called electrochemical cell. An electrochemical cell allows you to measure the so-called reduction potentials of molecules. Those reduction potentials tell you about the direction of electron flows in coupled oxidation-reduction reactions. So electrochemical cells can be constructed by linking two redox couples together, such as the zinc and the copper system. Here's our first attempt to build such an electrochemical cell. The cell is composed of two electrodes, one zinc electrode and one copper electrode. Both electrodes are submerged in one container that includes a solution of copper sulfate and zinc sulfate. So it's a mixture. The ions are all over the place now. The two electrodes are connected via a cable so that electrons can flow from one to the other. And then there is also a voltage measurement that takes place here. So let's take a look at what happens at the zinc and copper electrodes in this configuration. So as we already understood, zinc will be oxidized and go into solution as zinc 2 plus. Now, two electrons will be made available from that. And those two electrons, actually what they do simply, they will just go to the copper 2 plus cations, which then form solid copper on the electrode surface. So there's not really a reason for the electrons that are generated by the oxidation of the zinc to go through this cable to the copper electrode, because they can just directly go to the copper ions that are around the zinc electrode in this bath. So no current will actually flow in this system because the oxidation reduction reaction happens just on one electrode, the zinc electrode. So we have to refine our cell a little bit. Let's separate the two electrodes into two separate containers. The first one on the left now has a solution of zinc sulfate, and here we will have the zinc electrode submerged. And the second container will have a solution of copper sulfate, and here the copper electrode will be submerged. So in principle, now the reduction of the copper cannot happen anymore at the zinc electrode as it did here, but it must happen at the copper electrode now, so that the electrons from the zinc oxidation, they need to travel through the cable into the copper electrode to reach the copper that is hungry for these electrons, it wants to be reduced. But there's still a problem with this cell. It's still not going to work. The reason is, when the zinc metal now becomes zinc 2 plus, then in the solution there must be electroneutrality. There must be as many cations as there are anions. But there's only a certain amount of these sulfate anions. And if now more zinc 2 plus cations are formed, there will be an excess of positive charge in the solution. And that cannot happen because of electroneutrality. The same thing would happen in the copper, near the copper electrode. If copper 2 plus cations would now be formed, would now take the electrons and become solid copper, then there would be an excess of negative charge. So here would be an excess of positive charge, here would be an excess of negative charge over time, and therefore the reaction would stop. And eventually, again, no current can flow in this cell. So we need to also now in this system provide a passageway for the sulfate anions, so that when more cations are generated here, they can be neutralized by sulfate anions entering in here. And that's realized via a so-called salt bridge, or this semi-permeable bridge. The bridge is made of a gel typically, or a ceramic material, which only allows the sulfate anions to go through, but not the zinc cations nor the copper cations. It's only permeable for the sulfate. So this way now the sulfate that is in the copper container can go into the zinc container and therefore neutralize the positive charge that is generated by the oxidation of the zinc. And we will see indeed a sustained flow of electrons from the zinc electrode to the copper electrode. So now we have successfully constructed an electrochemical cell that enables these two half reactions to occur on these two electrodes and that are connected via a cable that allows the electrons to travel from the zinc to the copper electrode. So generally, the way how this is always drawn is that you actually have the so-called anode, which is the negatively charged electrode, on the left. So in the zinc and copper example, the zinc electrode is the source of the electrons, and therefore we call that one the anode. Here is where the electrons come from, and this anode is drawn always on the left. It is the negatively charged electrode. The copper electrode on the right is the positively charged electrode. We call that the cathode. So here's a different way how to write these two half reactions, which is by separating them with such a line here, with the double line. And reading this is from left to right. That tells us that the spontaneous reaction from oxidation to reduction goes from left to right. But there is still something not completely perfect with our electrochemical cell here to measure the reduction potential. The reason is that this reduction potential, or the driving force for the current generation, it actually depends on the concentration of the zinc 2+ and copper 2+ in these two solutions. To measure well-defined reduction potentials, one has to do that at a well-defined concentration. And as usual, the standard concentration of 1 mole per liter is used here, both for zinc and copper in the solutions. But there's a problem here, because now if this electrochemical cell starts working and starts producing current, then over time the concentrations of the zinc 2+ and copper 2+ will change in these two baths, and 1 mole per liter will not be the case anymore. So we have to make sure that when measuring the voltage, that standard concentration of 1 mole per liter stays constant during the measurement. Only then can we say it happens actually at 1 mole per liter. And this is realized in the so-called, by measurements of the so-called standard cell potential. The standard cell potential is measured in such an assembly. So here you have additionally a power supply. The power supply, what it does, it provides exactly the same voltage that the electrochemical cell would provide, but in opposite direction. So it will actually resist the flow of electrons from, for instance, the zinc electrode by generating an opposite potential that stops the electron flow. But then you can measure with a resistor and that sliding connector how high that opposing potential needs to be to stop the electron flow, and then you can say this is the standard cell potential at 1 mole per liter, because now the electrochemical reaction is actually not really happening. There are no zincs being zinc 2+ being produced and coppers being reduced, because there's always this opposing potential which we can measure and then state as the standard cell potential at 1 mole per liter. That brings me to the summary of this clip. We've learned that molecules or metals have different affinities for electrons. These affinities for electrons are reflected in the oxidation or reduction, or short redox behaviors. We can use an electrochemical cell to measure the different redox abilities via a voltage measurement, as you have seen on the last slide. Next slide, we're going to go into more quantitative aspects of these measurements to understand how the voltage actually connects to the free energy difference between the two electrochemical couples. Thank you.